let's do this. 8.3. So essential idea, the big concept we need to keep in mind is that we take any kind of light, could be from the sun, could be from the light racks we have right back there in the room right now making or for our internal assessments. We have to take this light energy and convert it into chemical energy. So we're not changing the amount of energy, we're just changing the type of energy. Remember, kinetic, chemical, heat energy, light energy. There's a lot of energies. So for our body, what we need, chemical energy, what life needs. So we're going to look and see how that happens. Remember, this process is ancient photosynthesis, and it really is what has built the earth into what we see today. And remember, the chemical form of energy that we see is a type of sugar. And then life can take that sugar and convert it into pretty much anything else. So the nature of science, how science works. These researchers, I believe they were in Eastern Europe, came up with this way to make radioactive carbon-14. And then this other researcher, Calvin, figured out a way to use it to figure out the order and the products that are made in the Calvin cycle in the light-independent reactions. So that means, so basically one scientist made a discovery. Let's say it made an, oh man, my handwriting. Oh, gotta slow down. Oh yeah, slowing down helps. It's okay to have empty airspace. Discovery. That's one thing I'm figuring out on these videos. It's okay. Don't have to talk the whole time. In fact, good to have a pause and some silence. And then another scientist took that discovery and used it for a whole nother purpose. We'll say different research question. Because really, research questions, those are important. From your extended essay, from your IAs, very important. Well, let's get into it and see what we're really looking at here. Everything. This is the basic overall view. We're going to see a lot of diagrams like this in this 8.3 set of videos. The two sides here, we got light dependent reactions. It might make sense that the other side would be light independent, right? And let's check out the light dependent reactions first, since those happen first. And remember, they do take space inside these thylakoids. So when it says intermembrane space, we're looking at inside. We're looking at right here in this area. Okay, annotate along with me at home or in your car, on whatever, wherever you're at, in your plane. This is where these light independent reactions are happening. Out here in the stroma, it's kind of like if you look at this like a cell, this would be the cytosol, cytoplasm, and the stroma. That's where our light independent reactions take place. And I already mentioned this guy, Calvin. Calvin cycle is what we still use Calvin cycle for here. Like the Krebs cycle is also called the citric acid cycle, since that's an important molecule that gets recycled through it. But for some reason, we still call it Krebs mostly. Calvin cycle is usually referred to as the Calvin cycle. The stack of those thylakoids, I love that word thylakoid for some reason, is called the granum. And you do have two membranes. You have an outer and inner membrane in your chloroplast. And in this topic, we don't talk about what happens in between, and I'm sure something happens in there, but I haven't learned about it. So I wonder if there's something that goes on. Why there are two membranes? Probably there were two bacteria that came together, and then that got absorbed and gradually took over, so there's two, but I don't know. I gotta find that out. Discovery is awesome. But let's look right away. This first molecule that goes into the thylakoids is water. Remember, you got to give your plants water. This is why, for the light-dependent reactions. And what comes out of this is oxygen. Okay, and then it gets released. So the air, the oxygen we're breathing in the air right now. Remember, about 21% of the air is oxygen. Is from this process. Somehow, some way from this process. It's photosynthesis. And over here, we know that plants breathe carbon dioxide, right? So that's where the CO2 is involved in the Calvin cycle. And from the Calvin cycle, it basically uses up energy and gives basically ADP and NADP. And then using the energy from the light reactions, which we'll get into momentarily, ATP is created in another molecule called NADPH. Remember, these the ADP and the NADP get reduced, right? Because reduction reactions build molecules, right? Add hydrogens, add electrons. And that's what we see here, which means there's some molecules that got oxidized. And then we see these redox reactions in the Calvin cycle as well. Well, let's move on to the next one. Let's see. We see right at the top, light. Another diagram. I added just, I don't know, another way to look at it. Light, we're taking these soon to be reduced molecules and reducing them, building them into these energy containing. So energy carrying. Remember, there was that little cartoon in the last set of videos, I think it was 8.2, where the water is saying, hey, I'm sick and tired, or the NADH was saying, I'm tired of carrying all these electrons. The proteins in the electron transport chain are saying, yes, give them to me, I love them. So we see the NADP, ADP being reduced. Remember, that water is turned into oxygen and then released. And that's all this is happening. Most of it's happening right here. Okay, these light independent reactions right there, and the thylakoids. Now, I mentioned before, in the stroma, which is kind of like the cytoplasm, that's where we have these light independent reaction of the Calvin cycle. Remember, oops, fix that. There we go. Left it on the eraser. Called light reactions or light dependent. And I guess I was saying Calvin cycle, it is called the light independent reactions too, okay? Instead of just the name. Light. And you all know, we mentioned before, that oxygen. And since, look at it, 6-sided ring, got an oxygen here. You should always think when you see this on a test or in a diagram, this is a monosaccharide, probably a glucose. Okay, but could be a fructose, or it could be galactose. Yeah, it was tough. I couldn't think of that third that third monosaccharide. But these, actually start out as 3 carbon molecules, and they get formed into this glucose, and then usually into starch. Basically, whatever product the cell needs, it'll get converted pretty quickly. All right. So reduced NADP, remember, it gets reduced. These phosphates are added, and we see here, remember this is happening, here we have the thylakoid lumen. Let me switch colors. Go. And kind of like right here, right along this membrane, out here is the stroma, as it's written right here. And this NADP is coming in off of the Calvin cycle, and it's getting turned to NADPH. Getting that NADP recharged, basically. And that ADP is turned into ATP and ready to go to the Calvin cycle, or the light independent, that is. See, it's so easy to mix up. So, word of advice, slow down when you're on your test or you're doing any kind of evaluation. All right, now let's get deeper into what happens during the light reactions. When you start looking at this, you're going to see, hopefully, you're going to see some similarities between photosynthesis and cell respiration. Because it's all about using proteins in a membrane to move electrons and using that energy to pull hydrogen ions across a membrane. That's what's going on there. So it starts out, step 1, there's 2 systems. And it's confusingly, the first one is called photosystem 1, and the second part is called photosystem. Sorry, the first one is called photosystem 2, and the second one is called photosystem 1. See, it's really confusing keeping that straight. So I'm not sure of a good way to remember that, but basically, light excites chlorophyll electrons. There we go. And they become energized. All right, and these electrons are coming from that water that's split. And they're coming up through the photosystem, and then they're going to dance along these membrane proteins. Given to photosystem 1, bounce along again, and we're going to make a bunch of stuff. Basically, as we do this, we're going to be pumping hydrogen ions across that membrane. Yep, hydrogen. We're going to be making NAD. This is kind of get ahead of my NADPH. And then these hydrogens are going to go through this ATP synthetase to make ATP. And then these all go on. But I'm getting ahead of myself a little bit. So going on there. All right, so here we got water, and so this is how water looks. It's a bent molecule. A couple electrons here kind of keep that shape bent like that, and it gets split. You'll see now after that light splits it, and really it's the chlorophyll that allows this to happen. Okay, that's the physical properties of the chlorophyll. That's the magic molecule here. I hate to use the word magic because it's physics. But it is awesome how this molecule came together in our natural Earth system to start this process, to allow it to happen, to give it this energy boost. Okay? And these electrons then get popped off this molecule, and then we'll go through these electron transport chains here. Like I said, it's going to pump these hydrogens. Going from in the thylakoid to outside. So they want to come back. We see, boom, come back. Remember, this part is going to make the ATP. Oh yeah. fill this out. These are electron carriers. And that's this this word photolysis is important. Remember, lysis means to split. Photo means using photons or light splitting. Okay, so this chlorophyll is a light splitting molecule. How cool. It's life splitting. So this is photosystem 2. Remember, that's the first part. And then again, we split that water, we get these electrons popped off, and then they're going to be moving through here. Ooh. Okay, and that's going to help generate our ATP later on, or actually this part here, NADPH. Very good. High energy electrons. There we go. And we got to deal with photosystem 2, right? So remember, you have photosystem 1. Those electrons get passed on from photosystem 2 to photosystem 1, where they kind of get re-energized. You can see them kind of lift up again into this electron transport chain. I keep remind myself to keep this straight. Remember, that makes the NADPH, those electrons moving through. Remember, this is light coming in here. Technically photons. This is all happening in the thylakoid interior space here. We kind of switched up. Usually I draw the thylakoid on the bottom, or the diagrams I use. Here this diagram is reversed. So it's kind of confusing, but then again, you know, what if on the IB exam they reverse it for you and you're like, oh, I didn't notice that. Remember, always read the diagram. So I did that on purpose. So thylakoids here, that means down here, this is where the Calvin cycle takes place. And you know it's the cycle. I'm just going to leave it like that. This molecule, I think I named it, maybe I haven't written it down. This is ATP Synthetase. Same thing you have, synthetase. Same thing you have in a respiration, right? Moving hydrogens from one side to another, generating ATP. It's very, very, very similar system. Okay, so at least there's that. Or out here, this is the stroma. All right, light independent. Just get, you know, fluent. Light independent, light dependent. You know, as best you can. It gets confusing. But you see, look at this big concentration of hydrogens building up. And then moving out and producing those ATP molecules. Awesome. I think I just realized earlier on, I think I was drawing hydrogens building up outside in the stroma, so that's incorrect. So just make sure, make a note of that. These hydrogens are building up inside the thylakoid. My apologies. So write that on your paper, maybe make a correction from earlier. Hydrogens build up inside thylakoid. So they go out, ATP synthase. And I see it written two ways. See how here it's synthase? It'll be synthetase sometimes. I think IB uses synthase. So I'm sorry, that is confusing. I'll try to just make sure I just use synthase. I know I make things more confusing sometimes. my apologies. My apologies. And we've seen this lots of times. NADPH now. Repetition is important to learning, as you know. And that's pretty cool. All together, we have the light hitting both these photosystems, both showing chloroplasts exciting these electrons. We see hydrogens being created, you know, from this movement of electrons and the splitting of water. Okay. Let's see. Movement of electrons and splitting of water. Oh, I did do it right. Splitting. Creating these hydrogens that build up inside the thylakoid, and then they need to go somewhere. Luckily, ATP has a nice little escape route for them, so they get shuttled right on out.